This is the structure of DNA. First thing to know is that there is this thing called the double helix structure. It was first studied by Rosalind Franklin, and she used crystallography to study the structure of DNA. However, Watson and Crick stole her work and used it to study DNA. With this, they found the structure of DNA to be called what is known as the double helix. Additionally, there's bases on the backbones of DNA. The backbones actually run anti-parallel, which is why there's a 5' end and a 3' end, and then a 5' and 3' end. The backbone has sugar and phosphate that make it up. Additionally, there are bases, like I just said. Adenine goes with thymine, and guanine goes with cytosine. Guanine and cytosine make 3 hydrogen bonds that hold it together, and adenine and thymine only have 2. Also good to know is that the number of adenines will equal the number of thymines, and the number of guanines will equal the number of cytosines. This is DNA replication. So first, helicase comes in, and it's an enzyme that unwinds the DNA. Next, I haven't drawn them, but there are single-stranded binding proteins that bind to the unpaired DNA strands, preventing them from coming back together. And they would be just holding these out. And then topoisomerase right here is an enzyme that helps to relieve the twisting strain, so it twists it. This allows it to be used as a template. So then primase will come in, and a short stretch of RNA called a primer is synthesized by an enzyme called primase. And then DNA polymerase, they break in new nucleotides and elongate the DNA strand. So DNA polymerase 3, as seen right here, adds new DNA to the 3' end of the DNA molecule. Oops, ignore that. So this will be 3', not 5'. To the 3' end of the DNA molecule. And the DNA polymerase 1 replaces an RNA primer with DNA, which is right there. For reference, anti-parallel strands would just be like this, so 3' to 5', and then 3'. It's just opposite, right, like that. I think I need to switch them, but either way, they're anti-parallel. Also to know is that there are leading and lagging strands. So in this case, this would be the leading strand because it's going into this. Like, it's the replication of DNA is going this way, which means that this is going against it. So then there's going to be Okazaki fragments, which need to be held together by ligase. And Okazaki fragments are present on the lagging strand.
So the reasons for cell division is for reproduction, growth, and repair. So the first step of the cell cycle is interphase, and this is where the cell spends most of its time. And there are 3 parts of interphase, which is G1, which is the first one. This is where the cell grows and carries out its normal functions. Then we have G2, which is where the DNA of the cell synthesizes and replicates, so 1 chromatid becomes 2. And then we have G2, which is where the cell finishes growing before it actually does divide. And so now we have our first step of mitosis, which is prophase. So the first stage of mitosis, the cell will prepare to actually divide, and this is where the DNA will condense to chromosomes, the nucleolus disappears, the spindle fibers will form, and the centrosomes move to either end of the cell. Step 3 is prometaphase, which is basically the stage in between prophase and metaphase. And so this is basically where everything that was happening in prophase finishes happening. So the DNA will finish condensing, the chromosomes, the rest of the nucleus will disappear, and the spindle fibers will attach to the connectors. Step 4 is metaphase. This is where the chromosomes will line up in the middle of the cell. Step 5 is anaphase. the kinetochore microtubules will shorten, and that will pull apart the chromosomes to either side of the cell. Step 6 is telophase. This is where the spindle or the nucleus for each cell forms, and then the spindle fibers will disappear, and the chromosomes will start to decondense. So this is basically the opposite of everything that happened in prophase/prometaphase. And the final step is cytokinesis, which is different for both plants and animals because animals or plant cells have the cell wall. So animal cells, the cleavage furrow forms, and it will pinch and divide into two cells. And in the plant cells, the cell plate forms as a pre-cell wall, and then they will divide. And that's it.
This one.
Yeah.
Oh, sorry.
That's okay.
So this is transcription and translation, and basically I'm just going to do like the differences between them. So transcription, transcribe means to rewrite. So what is it? I guess, so the synthesis,
Okay.
an mRNA molecule
Oh, veins. Oh,
From-
you first. Okay.
DNA.
What do you want to do?
And it happens because we need the information from DNA so we can express it in translation. So it occurs in 3 steps in the nucleus.
Yeah.
So we'll write that
Whoa.
in the nucleus. And the first step is
DNA.
initiation.
Wait, which one are you?
He's doing transcription, I'm doing translation.
Transcription.
So initiation.
There you go.
And in initiation, there is a promoter sequence. Transcription factors will bind to a promoter sequence, which includes the TATA box. Anyways, which are basically just chunks of thymine and adenine that are getting into the promoter sequence and are easily broken apart. The transcription factors summon RNA polymerase. Okay, RNA polymerase forming the transcription initiation complex. And then next is elongation.
There you go. Okay.
Elongation. And where the RNA polymerase checks down the DNA molecule using DNA as a template to make RNA. So I'll do DNA into RNA. Okay. It's unwinding the DNA as it goes, and the DNA will close back up after it finishes. And then the third step is termination. Termination. And it's different for prokaryotes and eukaryotes. So prokaryotes, I'm just going to write P for prokaryotes because I don't want to write all that out. So prokaryotes, the termination sequence is going to be transcribed. It's a nondescript thing, and it goes from prokaryote to prokaryote, which tells the RNA polymerase to disassociate and leave, and prokaryotes end here. I don't really know what I was going to write out for that, but okay. And then termination for eukaryotes, the polyadenine. I'm so sorry if I say this wrong. I'm just going to write it out because I don't know how to say it.
You know what? That's
So
right, that's your
the
synthesis.
polyadene, I don't know how to say it, adenylation signal sequence. Okay, signal sequence will be transcribed, which is the lab. Okay, which tells the polymerase to disassociate and leave the DNA. Eukaryotes also have another step, which is the modification of RNA. A 5 cap and a 3 poly A tail are added. The SNRPs, I'll just kind of write that here. SNR and P will form a spliceosome, which will cut out introns and splices together exons. And the end result is a mature mRNA. So that is transcription. And then translation. So the idea behind translation is to construct a polypeptide. Okay. Making a protein out of amino acids using the mRNA as instructions. So this occurs at a ribosome. A ribosome, and it happens because it's good to have proteins because they're needed to do a lot of things in the body and to basically live. So there are also 3 steps in this with an extra 4th step, which we'll get to. So 3 steps are the same as the transcription. So the first step is initiation. Initiation in which the mRNA comes together with a small subunit of the ribosome, and then it is then going to read the first codon, which is AUG. AUG bringing the amino acid methionine. Wait, I did not say that right. It's okay.
Methionine.
Methionine, thank you. Okay, methionine. GTP is then spent in order to bring in the large ribosomal subunit and form the translation initiation complex. I'm just gonna write TIC for translation initiation complex. And then next is elongation. Elongation, and there are three sub steps to this, I guess, which is the A site. And at the A site, which also stands for our rival, well, it doesn't stand for arrival, but we'll say it stands for arrival. The new tRNA arrives with the amino acid. The codon is read, and if it matches perfect, it was the right run, and it moves on. So then we have the P site, which is the polypeptide chain, and I call it the PP chain. Okay, and then the polypeptide chain is growing, and at the E site, the exit site, what is happening here is the new tRNA arrives at the A site. The chain from the P site is passed to the new tRNA at the A site. The ribosome translocates, which means moves down, putting the now empty tRNA at the E site, where it exits, putting the chain back at the P site, and now a new tRNA can come in at the A site. It's confusing, but yeah. Okay, the last step is termination. Well, not the last step, but it's basically the last step. Is termination. Which in one of the three stop codons, which are UAG, UAA, and UGA, will cause a release factor to bind, causing the translation complex to disassociate. Instead of bringing another amino acid, a release factor is brought in instead. So there is a step 4, which is basically just the protein folds. So protein folds in, now we have a protein, and that is transcription and translation.